Hi, my name is Jeane Thompson. I am a graduate student at Emporia State University, where Dr. Jensen and I have been researching whether or not behavioral tendencies present reproductive trade-offs in female dickcissels. So we are looking at phenotypic variation in animal behavior. Most people are already familiar with behavior. It is an action or group of actions in response to some stimuli, and they are influenced by both genetic and environmental factors. Behavioral tendencies, which we are interested in, are the consistent individual differences in behavior. You might think of them as an individual's average level of behavior. Behavioral types, then, are a specific combination or suite of behavioral tendencies. So if we're talking about aggression, then things like biting, orienting towards a competitor, and chasing them away would fall under this umbrella type category. We in particular are interested in two behavioral types, boldness and activity. Boldness is defined as an individual's reaction to any risky situation such as predation, and it exists along a gradient called the shy-bold gradient, where you have individuals ranging in very risky behaviors and low risk behaviors. Activity, on the other hand, is an individual's basic level of activity, and it exists along its own gradient where you have more and less active also as its extremes. Dickcissels are a common grassland bird. They winter in Central and South America, then migrate up here to begin their breeding season in mid-May. They do like to nest in grass clumps where they lay 3 to 5 blue eggs. They are polygynous, so one male may have multiple females nesting within his territory, but once the mating is complete, parental care is primarily done by the female. Dickcissels are also one of many species which are parasitized or brood parasitized by the brown-headed cowbird. So when we're looking at these relationships, we're not just interested in boldness and activity, although that's what we're talking about today. We're also going to eventually look at how they relate to nest depredation, brood parasitism, and nestling condition, which are three components of reproductive success, and we can see how that all relates and affects their overall fitness. So I have just spent the first of 2 summers collecting data at the Tallgrass Prairie National Preserve. We started in mid-May with the arrival of the male dickcissels. We found a total of 82 nests spread throughout our 3 different sites that we set up within the preserve. We found the nests by tracking females via their behavior back to their nests, or occasionally we would randomly flush one as we were walking throughout the prairie. Once we found a nest, we would monitor it twice a week until it either fledged or failed. We observed their behavior and recorded the nest contents. Each monitoring session consisted of 3 main parts: the preliminary observation, which lasted 5 or 15 minutes, depending on whether or not the female was actively incubating. We measured chip rate, perch height, so whether she perched above, at, or below the grass canopy. Her mode of nest entry is either above or below the grass canopy, and the proportion of time she spent visible throughout the preliminary observation. These 4 measurements we considered measurements of boldness, but we also looked at rate of movement, interactions with other conspecifics or other dickcissels, and provisioning trips made to the nestling by the female. These ones we considered measurements of activity. We then approached the nest and flushed the female, which enabled us to obtain the flight initiation distance, FID, and that's how close the female was willing to let us get before fleeing the area, the distance that she flushed from the nest, and her post-flush behavior, or PFB, which we ranked from 1 being the least risky behavior and 5 being the most risky behavior, such as a distraction display. These, again, are also measures of boldness. Once we left the nest area, we started our secondary observation, where we recorded the same 4 measures of boldness, but we also added latency, which is the time it took to return, for the female to return to the nest after being flushed. So our secondary observations ended as soon as the female returned to the nest, or we gave it a maximum of 25 minutes if she did not return. So our activities then, we looked at just rate of movement and conspecific interactions within the time that it took her to return to the nest. So if we're looking at the correlations now that we found between our secondary and preliminary observations, these are the measurements that 1 measurement was taken during the preliminary observation and 1 was taken during the secondary observation. So for instance, all of these are positive. So females who spent more time visible in the preliminary observation also on average spent more time visible in the secondary observation, and so on and so forth for each of these measurements. But we also, on the right here, we have the measurements that we took at the nest, and were only measured once for each monitoring session. We found a couple of correlations with those as well. So for instance, females who tended to flush shorter distances after fleeing the nest had more risky post-flush behaviors and shorter latencies. If we look just at the preliminary observation correlations, we see that they are, for this one, they are all positive correlations, and we see some interesting things. So females who were more visible in the preliminary observation also chipped more in the preliminary observation. They tended to perch higher, they moved more, and they interacted more. Females who chipped more during the preliminary observation also perched higher, had riskier post-flush behavior, and so on and so forth. In the secondary observation, we see not only a lot of the same correlations or similar correlations, really, but we see more correlations. And this is probably because a lot of these measures are measures of boldness, and in the secondary observation, this has taken place after we've approached the nest and looked in it, flushed her off, so we've become an active threat. And that might be why we are seeing more correlations in our secondary than our preliminary observation. So what we've done for all of these correlations, we performed the Kendall tau. So in blue, those were all our tau correlation coefficients. Red were p-values. We did Kendall because our data was not normal and had ties, but we found 3 main patterns. The first of which is that females who were more bold in one context were always more bold in another context. So if they had a very short flight initiation distance, if they allowed you to get very close, they also had very risky post-flush behavior or something along those lines. It was never a bold behavior correlated with a shy behavior. The same goes for activity. The more active a female was in one context, the more active they were in the other context. And the most interesting was our third pattern that we found, where females who tended to be more bold in one context were also more active in another context. So this is hinting at what might be a possible behavioral syndrome, which is when 2 behavioral types correlate with one another, but we won't know more about that until we continue our data analysis. So our next step is to use multi-factor analysis to create composite scores for both boldness and activity. We will then compare those composite scores with our reproductive indices. So thank you so much for listening. I hope you enjoyed this as much as I enjoyed doing it. I'd like to thank my committee members, Dr. Rebar and Dr. Ambadar, as well as Dr. Martin, for helping me on this research, my research technician who worked very hard this summer, and of course, Emporia State University and Tallgrass Prairie National Preserve for giving me the means to do this research. Thank you so much and have a great day.